Thank you very much, Martin. So thank you, everybody, for coming. It is a real pleasure to be able to talk to you guys about this project. This is the first time I've gone public with it. And it's a project that we started in late 2019. And I think that it is appropriate for this audience because it is, as I had mentioned during the pre-time, that this is a project that we're working on that is working strictly out of human specimens and trying to understand the nature of prostate cancer in that context, in the real context. Let me see how I can make myself forward here. So this project started, like I said, in 2019, and it started because of these two people. So I've been working with Riz and Felipe for several years now, looking at biology and biomechanics around failing hip implants in patients with metallic prosthetics, and looking at the formation of adverse local tissue reactions and the role of cobalt in driving that reaction through interactions with synovial fibroblasts and the stress responses that they impart based on that. And Felipe is a dentist by training with a material science and a histology background, and he is a gregarious individual. And many of you may already know him because of that particular aspect of his character. And he has expertise in microscopy, expertise in materials world. And working with Riz and working with me, we were able to put out several nice papers, and there's one more still floating around. And so we decided what we wanted to do after Felipe, after he earned his dentistry degree, earned a Ph.D., and he wanted to do a postdoc. And so I was really happy that he was interested in considering oncology. That hadn't been something that had been in his worldview. And so he has joined us, and he was trying to find a way that we could interface the different ideas that we already had some expertise in as it related to prostate cancer. And we chose this bone metastasis as a direction we wanted to go. And so this is stuff you guys all know, and I just want to make sure we're all on the same page. Prostate cancer is a major problem. The real issue in the modern world is that you guys do a really good job of treating these men. And so 95% of men diagnosed are living at least 10 years, but they're being really heavily treated. And the morbidities associated with those treatments are profound, as I have personal experience. And an increasing number of these men are developing metastatic castrate-resistant prostate cancer. And as Martin alluded to, prostate cancer has a high avidity for the skeleton, for the marrow-producing skeleton of the hip, the spine, and the pelvis. And so these are areas that decorate pretty heavily with the disease. And while this disease has historically been considered to be osteosclerotic, the clinical reality is that these lesions are mixed blastic and lytic lesions. And as you guys are aware, they are at high risk for fracture and spinal cord compression. And these morbidities really result in intractable pain and difficulty in management. It's really, really quite something. And so we want to try to understand that and see if there's anything we can glean from the kinds of studies we do that might help somebody somewhere down the road. And so we're not this old, but Martin was referring to Leland, and Leland is why I'm here. And Leland really beat this into me when we were at Virginia, the seed and soil hypothesis, Paget's work, really appreciating in the late 19th century that secondary growth of tumors are not a matter of chance. And that it really was a real question for us to understand what decides what organs shall suffer the dissemination of cancer. And what he really also appreciated was that the sites of metastasis are not passive in this. They're active participants in the metastatic process. And so this interplay between the cancer cells and the host are really interesting, really important, and really complex. And so we're finally at a point where I think we can really make some headway into this. If you read around, you'll see stuff like this. This is actually a recent cartoon, and you'll see ideas that look like this, where prostate cancer cells are shown in a bone niche where they had migrated out of the circulation. And what's come to be appreciated is that before the tumor cell ever gets there, that there are signals being sent systemically by the cancer, and extracellular vesicles are accused of being these messengers. And these extracellular vesicles can disseminate and help train the various organs that the cells migrate into. And in prostate, this has been really thought to be associated with changes that are involved in osteoblasts maturing to be cancer-associated osteoblasts, which is a very similar hypothesis to the idea that prostate cancers alter the activity of surrounding mesodermal cells, stromal cells, and that there are cancer-associated fibroblasts that have distinct phenotypes because of their exposure to these cells. And I don't want to get into that today. And we know that the prostate cancer cells produce factors such as MCSF, rank ligand, and other osteogenic factors that activate and maturing osteoclast out of the monocytic lineage, and this increases the mining of the bone, and that the class then produce other factors that stimulate the osteoblast to lay down more bone material. And then there is this vicious cycle of factors being produced, released from the mineral matrix, produced and released by the bone in resident bone cells that drive the prostate cancer cells to grow. Along the way, the prostate cancer cells practice a phenomenon considered to be osteomimicry. They produce factors such as osteocalcin, alkaline phosphatase, and bone morphogenic proteins that make them look a lot like osteoblasts, and they're considered to be ways that these cells help defend themselves from the rather adverse conditions of that bone environment where you basically walked into the garrison. That's where the immune system is, and they should be able to kill them right away, and they don't. So why don't they? So prostate cancer is not a singularity, as you guys know. It is an ever-changing spectrum of disease. And so this is a paper that came out of the Fred Hutchinson Cancer Institute several years ago now, describing from their rapid autopsy program the spectrum of AR states in their rapid autopsy program. And you can see that there has been quite a drift from the pre to the post androgen receptor targeted agent era in the percentage of patients that are succumbing to disease that represents an AR null or an AR independent characteristic. So we have neuroendocrine disease, we have double negative diseases, and this is an increasing population. And we honestly don't know anything about how these different disease states impact the various metastatic sites that are being affected. We really just don't know. It gets more complicated than that. Mark Lebrick, who is a graduate of Simon Fraser University, worked with me a bit with a guy named Tim Beischlag over there, and went down and joined Colm Morrissey and did a great postdoc. And he took that rapid autopsy program and further delineated using transcriptomics the spectrum of phenotypes of treatment refractory metastatic prostate cancer. And he broke it into three basic groups. These androgen responsive types of groups, the primary disease, AR high that develops in response to prolonged androgen deprivation type therapies, and African cells that appear to maintain AR, but eliminate things like REST that are associated with developing small cell disease. That there are under pressures of maximal androgen blockade, production of androgen receptor low cells that then have the capacity to potentially morph into various other AR null populations, such as small cell or NEPC, double negative populations, squamous cell carcinoma. So this is a complex problem. And do any of these actually affect bone in a different way? Are they all the same? We don't know. So that's what we want to know. That's the goal of the whole project, and I don't have the answer today. This is a brand new project, and we're just getting started. So as Martin and I were discussing before the talk, this has been work that's been going on for many decades since either of us were in the disease. And lots of prostate cancer cell lines exist, and people have been putting them into mice in various ways for many, many decades. And so the summary here is that from this data, there's not any way to really conclude whether any type of phenotype of a prostate cancer cell really gives rise to a specific type of lesion in the bone. So this is an example of data that comes out of the Hutch work from Ava Corey. And there's a reason why I'm talking about those guys so much. I'll get to that in a second. But we could see mixed lesions from LNCAP. We can see really overt blastic lesions from LUCAP. Other models that they have can be mixed. PC3 is typically very, very lytic, but you can read through all these things. You can see that different models present different things, not necessarily following any trend with AR activity or adenocarcinoma or non-adenocarcinoma status. And so there really just isn't a real consensus about what goes on. There have been a few more recent studies, so fast forward a decade or so. And one of Leland's models is C4-2b, which had a propensity to home to bone when injected into mice. In this case, was injected directly into femurs of mice. And again, you can see this sort of mixed phenotype where you get sort of blastic lesions that are a very abnormal bone formation and evidence of osteolytic lesions throughout this bone. And in this study, they were using Raman spectroscopy to try to measure changes in the material of the bone matrix. And they saw some subtle changes in a variety of components. They also looked at collagen mineralization, carbonate substitutions, and they saw some changes in these versus the various controls that they had. And this really represented the state of the art for what happens to the bone material in prostate cancer affected bone. Another paper came out more recently that used a variety of other techniques. Again, now this time using this first model, this PCA2B model, which was presented initially as being a very osteoblastic lesion, and in fact is still a very osteoblastic lesion, as you can see by these cross sections of these tibias. And so these are again mice. And what they were able to do was use some polarized spectroscopy to look at collagen networks in these bones. And what they reported was that there was really quite a distinct disorganization of this fibrils in the bone of the cancer-associated material. And that based on these sorts of fibril changes, they were able to measure a variety of physical changes to the bone, differences in Young's modulus, toughness, stiffness, just strength of the bone. And they proposed that the cancer-induced osteoblastic activity is disorganized, forms a more spongious bone versus lamellar bone, that results in an altered mineralization process and altered ability to develop normal hydroxyapatite formation in these bones. So in man, this is really the one and only paper that we're aware of where anyone has taken any kind of sophisticated view of what happens to the bone matrix of human vertebrae with prostate cancer. And so in this study, they have, count them, one prostate cancer patient, one. And they had a synchrotron, so it was early on the days, and so they were really applying that. And they had a handful of controls and osteosclerosis patients. And so this is their synchrotron three-dimensional reconstruction of what the bone looks like in this vertebrae of a patient with normal bone that got extracted for whatever reason. And this is the example of one region of the vertebrae that they had from the patient with prostate cancer. And you can see again that there's clearly an osteoblastic lesion and that there are residual trabeculi and a bunch of darker material that looks like bone matrix that is not well organized at all. And in a two-dimensional kind of slice, you can see these sorts of patterns. And so they wanted to measure what was happening with degrees of bone mineralization. And they were using the intensities of these reflected electrons to tell them what was going on there. And so they were looking at peak, mean, and width of these trabecular structures. And they reported that in the osteoblastic bone, there was a statistically significant decrease in these elements and an increase in the width. You see these very broad regions. And osteosclerosis also gets this very broadening, weakening of the bone. And so these things are not dissimilar phenomena. And how the bone resident cells that control these features are being protracted in prostate cancer to do something that looks like this, even way worse, really becomes the question. So those are the big questions that we're trying to answer in this first aim of the project. What are the alterations in the mineral composition and structure of the metastatic bone? And how do these lesions relate to bone weakness? So how are we doing this? Well, I called Colm, and who with Ava run the GU Cancer Research Lab down at the University of Washington. And they were really excited about the ideas that we were putting forward. They've been very helpful and helped us by providing us with our first 14 fixed mineralized bone specimens. Everything he had ever done was with demineralized samples. And so he had a handful of mineralized samples sitting in the archives, and he was willing to share them with us. And it was really nice of him to be able to do that. And of course, we have to thank all the patients and families of that rapid autopsy program. That's been going on for some time, thanks to Bob Vasella. And then we recruited colleagues out of Riz's lab. So Chun is a postdoctoral fellow who is expert in scanning electron microscopy and all things spectral. Dan May is a research operations manager at Hip Health, and she has done all of the MicroCT primary work for us. And then we've had the opportunity to recruit a couple of really energetic, imaginative, and bright undergraduates to work with us as co-ops. And so Samuel Zhu has worked with us along with Miriam Satra. In fact, Miriam was the first one in and came in during the COVIDium and made sure that we kept going. It was really great to have her there. And Sam was a house of fire when he was with us. So they deserve all the credit. All I get to do is talk about it. So what had Colm done? So Colm had published a paper back in 2012 looking at demineralized bone from this spectrum of patients. And he said that most of them are blastic, but every patient is mixed, and that you can find lesions that are blastic, that are mixed, that are blastic lytic, that are mixed, that are lytic blastic, whatever you want to call them. And there were really only a couple of patients that were straight up lytic. The rest of them are all blastic. So, you know, everything is kosher there. And out of this paper, he really wanted to say two things. First of all, there's heterogeneity of types within these various tumors. And second of all, ADT has profound effects on bone, full stop, and that these men are pinnic when they're on ADT, and the longer, the worse. And so bone health becomes a really major issue. And I hope that all of you guys consider that when you're dealing with your patients. And the last thing was, what is the effect of bisphosphonates? And so most of these guys have been treated with bisphosphonates before they died from first skeletal related events. And when he looked at the bisphosphonate treated patients versus the non-bisphosphonate treated patients, I'm looking over here now. So these are the differences. I'm not pointing it out, I'm sorry. So this is a more lytic region. This is a more blastic region. There's a real disorganization in the proteoglycan matrix, and under polarization, you can see that there are regions that are looking pretty well organized and areas that aren't. And then when he looks at the osteoclasts, he could see osteoclasts that were on bone just as well when the patients were on bisphosphonates as when they weren't. And so while there is some stabilization of the bone, it's not because they're kicking off or killing the osteoclasts. The osteoclasts are still trying to do their job. They're just not as good at it when the when the bisphosphonates are in there stabilizing that mineral. And so these are the 14 samples he gave us and what they had designated those samples as being. And so we have a core, they had a core. And what we found was that even within the cores that came out of the same vertebrae of the same patient, we didn't always score the samples the same as he did. So he had given us this information kind of as guides, and then we decided to compare it. So the reason they're split in two is because these 4 were non-bisphosphonate treated and these 10 were. And we haven't been able to find any useful guidance in whether they were bisphosphonate treated or not. So that still remains an unknown for us. And so this is how we characterize them. When I say we, this was Miriam and Dan May who did this. So Dan May did all the scans, and we created these 3-dimensional reconstructions of what these cores looked like that we had. And we scored these 4 as lytic. You can see these broken trabecular structures all over the place, some solid stuff, and occasionally these little clusters of aberrant formation of bone. So this is really what we're talking about. Thinned, broken trabeculae, all over the place, lytic. Then we have these 6 that we characterized as being generally osteoblastic with residual trabeculae. And this is a little designation that will show up again and again for this. And so you can see there's some variation in there. You can see some that are really nicely formed and others that are really pretty solid looking. And then you see these kinds of structures too. And so I'm going to focus on those. So really we see in 2 types that there are samples where there are residual trabeculae and what we think is new bone being laid down on top of that trabeculae, which is the ideas that Colm was trying to develop. But we also see regions that look like this under MicroCT, where there's what appears to be pre-existing trabeculae. In fact, there's more of it. But in addition, there's a bunch of matrix, a bunch of mineralized material that's in between the trabeculae that's filling these voids where the cancer cells are and is really looking very disorganized. And then we have samples that look like this, that are osteoblastic, but with little, if any, residual trabeculae. And in-house, we euphemistically called them marshmallow, and that stuck. And so I'm going to use that phrase today and hopefully never again. But this is kind of the way we look at it. And basically, they're dense, packed bone, all disorganized under MicroCT, but with no real structure. And there's one sample called 740 that we use a good bit because it looks very disorganized, almost completely as if the old trabeculae were all absorbed and new material has been laid down. But in it lies one little residual trabeculae that Felipe has found to be very useful as a guide. So Sam took these stacks of 3D MicroCT data, took a bunch of slices randomly through them, and analyzed a bone volume over total volume, and then analyzed them for these samples, and then plotted them out. And it was, in Alex's words, a sanity check, because the osteolytic ones all showed up as being low. That was good. And then the marshmallow and the osteoblastic with residual trabeculae were spectrumed out like this. And I didn't actually know how it would go, but it's actually intriguing that there's at least a trend where those that have appeared to have absorbed all of the pre-existing trabeculae and laid down new material are actually a little more denser than the ones that are still in that process. Is that a continuum, or are they distinct states? We don't know. But these are the kind of ideas that we'd like to pursue as we gather more samples from the hutch and start looking at samples that we are collecting locally to be able to compare directly to the kinds of cell environments that are associated with these lesions and try to make sense out of what's happening with them. So Felipe is, as I mentioned, a microscopist, and he likes to use different dyes and polarizations to look at different things. And so he's taken these samples, and so we're really focused on two types of samples, these samples that are this mixed residual trabeculae and new material versus these marshmallow-like samples. And he's used a variety of stains, in this case, Cirrus red and Mason's trichome, to look at collagen structures. And so you can see under polarization that in these trabecular regions, the collagen is well organized relatively. And then there are regions where the collagen is just not well organized at all. And both of these are basically saying the same story. Whereas if you look at this little region where you've got this bit of trabeculae, you can find regions where it is organized, but then it just all goes sideways. And you just have this disorganized material everywhere. And so clearly this is not structurally sound material. If this was your concrete, your house would fall down. So this is, a region that we're most interested in trying to understand, and how are these various forms of the bone structurally related to various stresses. And we're bringing Riz in to be able to do some of that work as we speak. The other thing he's done is looked at proteoglycans. And so proteoglycans are really important in mineralization of bone formation. And in this case, he's used toluidine blue just as a general stain for these polyanionic proteoglycans. And then he's used decrin as a specific proteoglycan associated with mineralization of newly laid down collagen fibers in bone. And you can see that there are distinct regions where you have these more residual regions, and then you have these darker regions where new material is being laid down and hasn't quite ossified yet. Here's another structure here where here's, again, this residual piece that Felipe uses as a guide, and then all this other material being laid down in this very bizarre network that has no rhyme or reason and certainly no structural integrity. And these are just parallel sections of these specimens. And you can choose decrin as a specific proteoglycan. So we thought, wow, maybe we can use these densities between what we consider to be the residual material and the new material as a guide to come up with some parameters that we can apply in an unbiased way to samples to say, where is the quote unquote new bone and old bone? And look at the densities of each one individually and that sort of thing. And so Sam likes to play with numbers, and he was willing to play with image J a good bit. And so he started coming up with ways to use image J to develop masks to define various areas of various intensity and looking at lots of regions of interest. And when he looked at these two specimens we were most most interested in playing with. We saw these various differences. They're easy for our eye to see, but we wanted to know if a machine could find the differences. And so these are the two specimens we were working with. And we were really excited to see that the lamellar bone that we could find from these two specimens showed a much higher calcium peak weight by X-ray diffraction from these samples out of these quantitative backscatter images that Chun had. And that the disorganized area was much lighter. And so we were hoping we would be able to do this over a bunch of samples and come up with some metric to have a cutoff and say, brightness above this represents lamellar bone, brightness below this represents disorganized bone. And so Sam got all excited about it. We said, okay, start doing this to a bunch of samples. And so he went at it for a few weeks and we had a meeting and we got this. And so the whole story blew up in our face because we don't show the sample we just showed, but the samples here, it's just all over the place. There's no way to make a call based on this. So we're still thinking about this idea of how to pick a specific type of component, calcium or some other heavy metal, heavy ion in the bone matrix to use as a guide to help us decide what is going on here. But basically the story is that using these sorts of calcium peak densities, we're not going to be able to make that distinction. So hypothesis one is gone. However, when we were looking at these samples, what we noted was that we kept thinking there was a difference in the density of lacunae between this residual trabecular bone and what we think is this new bone being laid down. And you can see these little trabeculae, and they tend to be a little different shape. So as is traditionally described in bone, these lacunae tend to be elongated, flattened as the bone material flattens out and becomes laminar. And that in the newly formed bone, where these osteoblasts or osteocytes now are doing their job or have died not doing their job, depending on your perspective, that they're rounder. And so Sam came up with a mask system, again, using image J, and we defined some upper and lower limits on how big a black spot could be that we would call a lacunae. And so he went out and he started scoring a lot of samples. And so this is the table over here on the right that shows the list of all the samples and how we had defined them, and then a real absolute number, what was the lacunae density in the regions of interest that he was scoring from these different regions. And this is how it plots out. So it's not the best in the world, but certainly we can see a distinction between the disorganized and the lamellar bone. The lamellar bone has fewer, tighter density of lacunae than the disorganized bone. And so again, within these few samples that we have so far, this is the kind of density that we have. And so we ran out of time with Sam, and we'll have to continue to use his programming expertise to develop this further with the next round. And we will go ahead and finish the spectrum of 14 samples, and hopefully we'll be able to get the rest of, if not at least another batch of 14 samples from the Hutch for their fixed specimens. And if need be, we'll start to pursue other rapid autopsy programs around the world to see if we can't come up with more of these fixed, non-demineralized samples. Because we think that this will be a helpful way for us to try to distinguish the kinds of bone that we see in a more arbitrary way, be able to really define them without any kind of just it looks good. We want to be able to have a number that we can put on it. We think we can use these various approaches of proteoglycan and these sorts of densities to be able to distinguish one type of bone from the other, and then apply that to the new material that we're collecting out of specimens that are coming from our local surgical community. That brings me to part 2, which is the work that we've started here using specimens that are being retrieved from patients that are undergoing surgeries for compression fractures of their spine, and hopefully soon from revisions of hip from men with infected or involved hips. And so this started when Felipe was talking to Antonio, who was doing tissue collection for us, and talking about our project. And Antonio said, go talk to Rafael. And so he did. And Rafael jumped in with both feet and has been a really active member of the team since we first started talking to her. She's coming to our group meetings and really participating and really interested in trying to find ways to bridge the gap between what she does as a surgical spine oncology expert and what we do here as a biology program. And again, we have to thank all the patients and families of the Vancouver Spine Surgery Institute for consenting to allow us to use the samples that we collect from these patients. And then we're working closely, of course, within the VPC with Dr. Collins and his Laboratory for Advanced Genomics Analysis. And I really want to point out the expertise of Anne and Funda, who have really made our ability to try to do single-cell sequencing possible. So our questions here are, can we link the types of bone lesions with cancer genotype or phenotype? What changes to prostate cancer cells occur in the bone niche? Unknown. And how do metastatic cells affect bone cell control of the abnormal matrix formation? So these are the overarching questions that I have no answers to yet, but that's where we're going with this. And so the first specimen, Felipe went over and basically was viewing a surgery that Raphael was doing. And there was a spinous process off the backside of a vertebrae that was involved in this particular compression problem. And it hit the trash. It was of no use at all. And Felipe grabbed it. And we had permission to be able to do that. We worked it through our biobank here. And he brought it back, and we got that little piece of bone on the MicroCT, and we got some analysis at MicroCT, and we were able to see what looks like normal looking bone in that spinous process, and then a bunch of stuff that looks like it might be some of that new bone that we've been talking about. And in a section related to that was demineralized, Felipe was able to see that there's some cells in there. That was always good to see. And maybe some evidence of some old bone and new bone. And when you zoom in a bit, you can see that there's this highly refractive residual bone. And then again, these layers of darker material that we suspect is new bone being laid down by this complex things that are happening around there. I don't know what just happened on my window here. Anyway. So we have so far acquired 2 specimens, and those specimens are related here. So these are Raphael's x-rays prior to surgery of the vertebrae we're going after. And so from these specimens, we see 2 different types of formation of cells within these bone lesions. And so we see them both in each one. And so we don't have enough samples to actually say, well, look at sample 1 versus sample 2. So these are just samples from these 2 patients, where you see nests of cancer cells embedded in a bone matrix with a very distinct region around it. And then you see areas where there's this complicated trabecular structure, new bone material being laid down, and cancer cells just decorating and resident bone cells just decorating the space everywhere. And we've been working with Larry and Nathan on some ideas about targeting PSMA. And so we have some decent reagents for PSMA. And so Felipe applied some of those to these specimens to try to define who was prostatic and who was not. And so in these nests, you can see there's nice decoration with PSMA on many of these specimens. And we also see some in these other areas, and areas where there are cells that aren't positive. And what we find most intriguing is that in these nests of prostate structures, that they take on what could almost be scored in a Gleason grade. And they form pseudoacinar kinds of structures. And so it would appear that the PSMA is decorating exclusively on what we would consider to be the luminal face of these cells. And not all the cells are positive, at least in this particular section. But we see these nice luminal expression. But we also see cells that are not in these structures, that are just spread out all over the bone matrix. And they don't seem to have any kind of apical polarity at all. And so they're putting out PSMA and staining on all surfaces with their PSMA. So these 2 different characteristics. And are they the same types of cells? Are they the same genotype, the same phenotype of cell, or are they different cells altogether? And we'd like to be able to get at some of that information. And one way we know how to get at that kind of information is through single-cell sequencing. And so we got a sample from patient 3 who presented as a naive patient with advanced metastatic disease. And so it's a unique opportunity probably in the modern world. And Felipe was able to get some of that material and the Laga group was willing to drop everything and help us process that sample because we haven't figured out how to freeze and collect nuclei yet from these to try to do those. That's a goal of ours. But this was sample we were able to run with. And we didn't know anything about how to break up the bone and get the cells out. This was just blind. And Felipe and Anne and Funda really went at it, and they pulled it off. And it took a few things to try, but they pulled it off. And so we didn't get the best sequencing depth and the most cells ever. We're not trying to say we've broken the world record on anything, but we have actually got it done, and that's the key. And from this sample, we analyzed gene expression signatures from, I don't know, about 1,000 cells, a few thousand cells. And then we use these Tinsey plots that are basically complex t-test analyses between various samples, and in this case, thousands of samples, looking at thousands of genes. And then this is a graphical way to display the overall relationship in a two-dimensional space of the different types of cells that you identify using these sorts of sequencing approaches. And so we see clusters of cells that look like they're prostate cancer. We see traditional adenocarcinoma cells that we consider AR high, PSA high. We see AR low, PSA low cells that are also very prostatic. And we even got a couple of cells that we scored as prostatic and neuroendocrine based on chromogranin staining and other markers within that population. We also see a group of cells that are considered mitochondrial high, which are often considered trash in these sorts of single cell sequencing assays, that they're stressed cells, cells that are dying or dead. And, but they link up with the prostatic cells. And so we wonder whether these are just cells under high degree of stress. Throughout the program, we're very interested in cell stress and how therapies induce these and how the cells respond to those sorts of things. And so I'm not going to take my eye off the ball. I'm going to keep my eye on these cells. I think they might be very interesting as they relate to the prostatic populations. We see some red cells, and then we see the resident cells all clustered together. And so we see CD4 and a huge population of CD8 T cells. Felipe and I have been very interested in T cell exhaustion and T cell infiltration in these adverse local tissue reactions. And so it'll be interesting to compare some of the ideas we have coming out of that project with what's going on here. We see monocytic cells, macrophages, and their relatives, the osteoclasts. We see osteoblasts, which have no real relative here and are kind of just stuck in the middle. We see populations of neutrophils and B cells. And so these are just first level characterizations. And as we collect more of these, we'll be able to start to talk a little bit more about what are the subtypes of these various leukocytic populations, and more to the point, what are the subtypes of these types of cells? And then the next level question is, how are these things spatially related? And so we would love to be able to pursue some spatial profiling of these different cell types within these lesions. And I think Felipe has done a pretty good job of showing us that that ought to be doable. So he's looked at a variety of things.
Can I ask a quick question?
Please.
So I'm sorry I joined late. I came from another presentation. The population here that's AR negative and KLK3 low,
they shouldn't be negative. They're low. They're not.
Okay. Because so I was going to ask what you determine as conclusive proof that they are in fact prostate cancer cells at all.
Right. So yeah. So they do produce some AR. They do produce PSA, PSMA. They produce the right side of keratins. There's a list of 14. I didn't bother to show that table, but it's a long list of genes that we use as to define that signature. I'd love to talk to you about that more, Alex. Okay. And then these neuroendocrine cells, they score with the traditional markers. They look just like what we were describing 20 years ago. So they're good. Okay. Gotcha. Okay. Yeah. I'm sorry. That's my bad. That was something we did, and I just never have changed it. Okay. Thank you. So Felipe is trying to use at least 2 colors at a time in conventional histology. And so this is a case where he's staining for PSMA in red, again, showing that the acini-ish structures show these luminal faces where that's where the PSMA is. And that we can see cells that are PSMA negative that are still cytokeratin positive. And, they should all be cytokeratin positive to some degree. And so we're working on these sorts of stainings. When we look close, we can see places where there are AR positive cells, AR negative cells, all in the same area. And what are the genotypes and phenotypes of these different cells? We think that the spatial profiling would be the ideal way to go about looking at some of these ideas, some of these issues. And up against the bone, we can see areas where there are what are clearly prostatic cancer cells, AR positive, pancytokeratin positive cells sitting on top of the bone matrix that is covered with a layer of cells that would be easily characterized as being osteoblastic, but as soon as we have a marker, we'll feel more confident about that, but that are forming these things. And so then what are the relationships here? And what's happening in this blast versus a blast somewhere else that's very, very different? How has it influenced the kinds of osteocytes that are deeper in the bone? And how are these things affecting the prostate cancer cells here and their structural organization versus the cells that are occurring in places like this where they're free and not on the bone material at all? So those are the things that we're trying to do. And these are the steps that we have so far. And we're really excited to be able to have the opportunity to pursue this project. And I'm going to close by thanking, there's clinical questions, all the things we want to know. I want to leave some time for questions, but is fracture associated with bone quality? And are these areas of bone resorption? Is pain related to fractures, microcracks, or bone strains? Probably some combination. Or is it caused by really just the soft tissue pressure on the periosteum and those sorts of things? These are the areas that working with Raphael, we're really looking forward to pursuing. Can we identify these different types of lesions in patients? Do patients with different lesions need different strategies for support? And can we quantify and identify those direct effects of maximal androgen blockade on the bone? Because I don't think we really appreciate the spectrum of things that happen over the decade or so that men are exposed to these sorts of things. And can we design strategies to reduce those effects? I think those are the things I'd like to be able to say we did out of this project in sometime before I retire, which will never happen. So there you go. So really, it's been an exciting road, and we did this on nothing. We did this on a shoestring. And we've worked really hard at it, and we're really, really grateful for recent support we've gotten from a number of agencies to put a project together that we think we can pursue for a few years. So the Canadian Cancer Research Society Inc. put up support for us as a research project. Michael Smith and PCFBC have provided support for Felipe and trainees. Fred Hutch put together support with us to support a pilot spore project. And we're really excited about using those funds to be able to pursue our single cell sequencing and cell type distributions within these structures. So that's what we're doing. And I'd be happy to take any questions.